Okay, the door is closed. Good morning, everyone. My name is Jury Huang, and I'm from Korea Bio. And looks like I'll be emceeing the session 2, keynote session, with Dr. Virginius at the last day of BIOPLUS-INTERPHEX 2022. We're very honored to have you as an audience for this and thank you for dedicating your precious time at the conference. And another person that I should be honored to introduce is Dr. Virginius Cas9, if I pronounce that right, from Vilnius University Institute of Biotechnology, who will be giving the inspiring speech on the past, present, and future of CRISPR-Cas technology. Dr. Cas9 is a world-renowned Lithuanian biochemist. He studied organic chemistry and obtained his master's at Vilnius University, where he is currently the head of Department of Protein DNA Interactions. And for his work, he has received numerous awards, including the Warren Albert Foundation Prize and the Novozyme Prize, just to name the few. Dr. Cas9's main research interests focus on the antiviral defensive system in bacteria, inspired by a 2007 paper that reported an adaptive immune system provided by the CRISPR DNA sequence in bacteria. He began working on understanding the role of enzyme Cas9 CRISPR. Ladies and gentlemen, let me introduce Dr. Virginius Cas9 to the stage. Please welcome him with a round of applause.
Yeah, so thank you very much for a kind introduction. It's my pleasure to be here. It's my first time in Korea, and I'm really happy that I already started making friends. So I bumped at Starbucks this morning into our presenter today, and actually she was working hard trying to pronounce my family name. It's not easy, yeah. So my lab is located at Life Science Center of Vilnius University, and of course during this meeting I already was asked several times where Vilnius is located. So I decided just to put a slide to show you. So Vilnius is a capital of Lithuania, and Lithuania is one of the Baltic countries located on the shores of the Baltic Sea, and Sweden is just across the Baltic Sea, and Poland is our southern neighbor. And Vilnius is a UNESCO heritage site, probably most famous for the number of churches per square meter. But this doesn't mean that actually we go to pray to church every morning. Just across the river, there is a modern city. And in Vilnius, history and modern times usually live together. And Vilnius University is one of the oldest universities in the region. It was established in 1579. And very recently, we moved into a brand new life science center of Vilnius University that is located at the new Vilnius University campus. And a few years ago, actually, Vilnius appeared on another map that I borrowed from the review of Eric Lander that was published a few years ago. And Eric actually called this map the CRISPR map of key discoveries, and I was very happy to find that actually Vilnius was on this map. So it's probably not that surprising today that I'll be talking on CRISPR. And I was asked to talk about past, present, and future of CRISPR-Cas technology, and since we are very, very early beginners of this CRISPR story, so I will talk about past, present, and future for CRISPR-Cas technology from the perspective of my own research. So in 2007, a paper appeared in a science journal that claimed that a new antiviral defense system called CRISPR was discovered. So actually, why these antiviral defense systems are so important in bacteria? So actually, during the pandemics, we all become acquaintance with viruses, and now everyone knows what that viruses infect humans, but viruses in fact also infect bacteria. And phages or bacterial viruses infect bacteria in order to replicate, and usually they kill bacteria when replication is completed, so therefore they represent a lethal threat to bacteria, but bacteria somehow survive. And this is because bacteria during evolution was building multiple defense barriers, and since phages need to get into bacteria in order to replicate, they are trying to break these defense barriers, and this forces bacteria to develop new defense barriers, and this kind of arms race between viruses and bacteria is going on and on. And in my lab, actually, we are studying different antiviral defense systems in bacteria. And there are really many different antiviral systems in bacteria, and the genes that are encoding different antiviral systems in bacteria, they are clustered in a so-called defense islands. And in these defense islands, genes that encode restriction modification system, CRISPR, and other antiviral defense systems are clustered together. And these antiviral defense systems are very important for bacteria. For example, in M. aeruginosa, nearly 30% of genome is occupied by antiviral defense systems. This shows that, in fact, in what kind of hostile environment this bacteria survive. And during recent years, in my lab, we focused on the CRISPR-Cas antiviral defense system in bacteria. So when virus infects bacteria, most of the bacteria die. However, a small number survive. And it turned out that survivors were able to hijack small pieces of viral DNA and insert them into a so-called CRISPR region that is present in bacterial genome. And then bacteria uses these inserted pieces of viral DNA as templates to make small RNA molecules that, together with Cas proteins encoded by Cas genes, make a so-called effector complex that is able to identify virus during next round of infection and destroy it. In this respect, CRISPR-Cas system functions as an adaptive immune system in bacteria, since it's able to memorize the invader by inserting pieces of invader's DNA into the genome, and later use this information to recognize the invader and destroy invader's DNA. So in my lab, we are trying to understand the molecular mechanisms behind the CRISPR-Cas antiviral defense system, and we are using a combination of different biochemical, structural, and biophysical methods to understand molecular mechanisms behind CRISPR-Cas systems. So we started by studying CRISPR-Cas systems in Streptococcus thermophilus bacteria, and Streptococcus thermophilus is a bacteria that is important in industry because this bacteria is widely used for production of cheese and yogurt. And in fact, this bacteria contain 4 different CRISPR-Cas systems. And for our studies, we decided to choose the CRISPR-3 system of Streptococcus thermophilus for two reasons. First, because this system contains smallest number of Cas genes in the CRISPR cluster, and next, that one of the genes, Cas9 gene, encoded a protein that had an active site that was similar to the active site of restriction enzymes that we seen before. So we didn't know how to work with Streptococcus thermophilus bacteria, so we decided to transplant the CRISPR-3 system of Streptococcus thermophilus into E. coli that was a working horse in my lab. And to our surprise, actually, the CRISPR-3 system of Streptococcus thermophilus was functional in E. coli, and it was able to provide protection against invading phage or plasmid DNA if a matching spacer was present in the CRISPR array. So then, using genetic tools available in E. coli, we were able to demonstrate that Cas9 is the sole gene that is required for interference. And actually, this experiment was the first demonstration that CRISPR systems are independent functional units that could be transplanted between different organisms. So, of course, the next logical step was try to isolate Cas9 protein from E. coli. And Gedros Gasiūnas, who was a PhD student in the lab at that time, was able to isolate Cas9 protein, and he showed that Cas9 protein actually co-purified with two RNA molecules, CRISPR RNA and tracer RNA. And later, we used a number of biochemical assays to demonstrate that actually the Cas9 protein, guided by CRISPR RNA, is able to locate a target sequence in the invading phage DNA and bind to it, making a structure that is called R-loop. And then Cas9 protein, using two different active sites, is able to cut DNA, introducing double-strand break. And a simple modular organization of Cas9 complex, where specificity is encoded by CRISPR RNA and cleavage is provided by Cas9 protein, set a stage for creation of universal RNA-guided DNA endonucleases. Indeed, simply by changing CRISPR RNA in the complex, Cas9 protein can be targeted nearly to any site in the genome where it will make a double-strand break, and then this double-strand break trigger mechanisms of the cellular repair that could be used for gene knockout or knock-in. And the easy programmability of Cas9 complex by customizable CRISPR RNAs made Cas9 a preferable tool for genome editing in very different model organisms. And of course, Cas9 is a great tool that is rapidly advancing into the clinic for treatment of different diseases, including eye disease, lung disease, liver disease, and others. However, there are still challenges that need to be overcome. And one of the challenges is targeting space limitation due to the PAM requirement of Cas9 protein. And in fact, I will talk about that a bit later. And another problem is related to the Cas9 delivery into the cell. Cas9 is quite large protein, and adeno-associated viruses, or AAVs, are preferable vector for delivery into human cells. However, Cas9 is too large to be packaged into AAV, so therefore smaller Cas nucleases or Cas9 variants are required. And of course, there are other challenges, but in my talk today, I will focus on the first two. And of course, there are two broad solutions to address these challenges. One of these solutions is just to use protein engineering, trying to modify existing Cas9 variants to overcome these limitations. And another possible approach is to use to look at the natural diversity of Cas9 proteins. And in my lab, actually, we focused on the exploration of CRISPR-Cas diversity. And CRISPR-Cas systems, in fact, are very diverse. And this phylogenetic tree just shows the diversity of CRISPR nucleases that include both Cas9 and Cas12 nucleases. And during last part of my talk, I will try to tell you three short stories. First, I will focus on exploration of national diversity of Cas9 orthologues. that helped us to overcome the PAM limitation problem. Then I jump on the miniature nucleases that allowed us to solve the problem of the delivery of nucleases. And finally, I will talk about novel nucleases that come from transposons and that are not related to CRISPR. So the specificity of Cas9 is dictated by interactions between CRISPR RNA and matching DNA target. However, to initiate binding of the CRISPR RNA to the DNA, Cas9 protein first has to recognize PAM sequence. And PAM sequence is a characteristic feature of each Cas9 protein, and different Cas9 proteins recognize different PAM sequences. And this PAM sequence requirement, in fact, limits target site selection. For example, the GG-PAM requirement for aspergillus Cas9 protein constrains targeting to every 8 base pairs in human genome, and this makes nearly 75% of mutations associated with human disease non-accessible if a precise targeting is considered. So we reason that exploration of the Cas9 diversity could help us to overcome target space limitation problem. And Cas9 proteins are really abundant. If you look at the databases, you will find thousands of different Cas9 proteins. And to identify new Cas9 variants, we developed a phylogeny-guided bioinformatic approach. And using this approach, we were able to identify thousands of novel Cas9 sequences. And actually, using this information, we were able to build a phylogenetic tree of Cas9 proteins. And in this phylogenetic tree, you can see that actually Cas9 proteins fall into 10 different clusters. And then, to sample the Cas9 diversity, we selected 100 proteins from this phylogenetic tree, and these proteins belong to different Cas9 clusters, and aim to establish their PAM requirements. So the genes encoding Cas9 proteins were codon optimized and expressed in the in vitro translation system. The guide RNA was predicted bioinformatically and also transcribed directly in vitro translation system. And the Cas9 guide RNA complex was assembled directly into in the IVT mixture, and it was used to digest the plasmid DNA library that contained a unique sequence targeted by guide RNA, and this unique sequence was flanked by randomized 7 nucleotide sequence that corresponded to the PAM position. And then actually we captured cleavage products by adapter ligation and deep sequenced them to establish PAM sequences that license DNA cleavage. And this approach allowed us to identify a really large number of new Cas9 variants, and using this assay we were able to determine, to show that nearly 50 Cas9 orthologues show new PAM requirements, and Cas9 orthologues recognize different PAMs that some of them were G-rich, some of them were A-rich, some of them C-rich, or some of them contained mixed nucleotides in the PAM sequence. And the PAM length was varied from 2 to 5 nucleotides. So the take-home message from this part of my talk is that actually Cas9 orthologues is a rich resource of potential novel genome editing tools, and these Cas9 proteins recognize around 50 new PAM sequences. And actually, the search for these new Cas9 orthologues is ongoing in Cas9 company, and those of you who are interested in these Cas9 orthologues have to approach them directly. So I just show you how exploration of Cas9 diversity allowed us to overcome target space limitation problem. In the next few slides, I will show you actually how exploration of natural diversity allowed us to overcome the size limitation of nuclease variants. And recently, Schmackhoff and Harrington identified a new CRISPR-Cas system that contained a so-called Cas12F effector protein. This Cas12F protein, in fact, is a part of the family of Cas12A proteins, so well-known genome editors. However, there is one essential difference, that Cas12F proteins are nearly 2 or 3 times smaller than Cas12 protein. So therefore, we call these Cas12F proteins miniature nucleases. So we selected a number of genes encoding Cas12F proteins from the phylogenetic tree, and then actually expressed these genes in E. coli together with CRISPR array targeting a specific sequence in the plasmid DNA library. And then we use the same assay to establish the cleavage requirements of miniature nucleases. And what we found that indeed all these miniature nucleases were able to cleave DNA, introducing double-strand break, and cleavage was dependent on the PAM sequence. And this, we were able to establish PAM sequence for 10 different miniature nucleases. Most of them required T-rich PAM sequence. However, a couple of them actually were specific for C-rich PAM. So next we decided to look whether these miniature nucleases could be used for genome editing in human cells, and we engineered a plasmid that contained guide RNAs that targeted 3 different sites in the human genome, and also encoded 2 different miniature nucleases. And human cells were transformed with these plasmids, and then actually DNA editing outcome was established by deep sequencing, looking at the indels at the target sites. And we were able to show that one of these nucleases, Cas12F nuclease, was able to introduce indels still at low efficiency, but it was the first demonstration that these miniature nucleases are active in human cells. And very recently, a number of papers appeared in different journals showing that these miniature nucleases could be used for different applications in different model organisms. And one of the papers came from the Korean group, 2 other papers from different labs, and our own paper showing that these nucleases could be used for plant genome editing. So in the next few minutes, I will show you a different story that is to somehow related to these miniature nucleases. And actually, these miniature nucleases, in fact, now that are half size, that are half size of the Cas12 protein, could be used for the genome editing applications in eukaryotic cells. And these miniature nucleases, again, are available at Cas9. So if you look at the phylogenetic tree of Cas12 protein family, you can see that these miniature Cas12F nucleases has a very close relative, a TnpB protein. So Kunin and collaborators, they predicted that this TnpB protein could be a progenitor of Cas12 protein family. So what is the TnpB protein? And TnpB protein, in fact, belongs to the transposon family that has this strange name, IS20605. And transposons are mobile genetic elements that jump between different locations in the DNA. And transposons of this family are probably one of the most simple transposons, because very often they are comprised of only 2 genes, TnpA gene and TnpB gene, and the transposon is flanked by left and right elements. And the TnpA is a transposase that mobilizes transposon. And transposon is excised during DNA replication, and transposon excision occurs through a single-stranded DNA intermediate. And then, actually, the transposon is excised close to the TTGAT sequence, specifically for the transposon that we studied. And then, actually, transposon is looking for another sequence, TTGAT, in the genome and inserts the excised transposon at this particular location. So the role of TnpA is well established. It's a transposase. But what TnpB was doing in this system was not clear. So we focused on a particular transposon that is present in Deinococcus radioadurans bacteria, and we aim to isolate TnpB protein. And we actually expressed TnpB protein together with inactivated transposon in E. coli and purified this TnpB protein. And it turned out that TnpB protein co-purified with RNA molecule, and then we performed RNA-seq to establish what kind of RNA molecule is it. And we found that actually this RNA molecule that co-purified with TnpB originated from 3' end of the TnpB gene, and also included right transposon element. And the last 16 nucleotides of 150 nucleotide RNA originated from plasmid DNA sequence. Since this RNA included right element of the transposon, we called it RNA or right element RNA. So next we decided to establish DNA cleavage requirements of this TnpB nuclease, and we again used the same assay that we used for characterization of Cas9 orthologues or miniature nucleases. So we expressed the TnpB protein together with rare RNA in E. coli, and the rare RNA actually targeted a unique sequence in the plasmid DNA library that was flanked by random 7 nucleotide sequences. And using this biochemical assay, we were able to demonstrate that TnpB, in fact, functions as RNA-guided DNA nuclease that cleaves double-stranded DNA at specific, when a specific TTGAT sequence is present in the vicinity of the target. But if you look at the transposon, so the same TTGAT sequence is present close to the left element of the transposon. So therefore, we call this sequence transposon-associated motif, or TAM. So taking together, this experiment showed that TnpB, in fact, functions as the RNA-guided DNA endonuclease that is able to introduce double-strand breaks in the DNA. And of course, the next obvious question was whether TnpB is able to perform gene editing in eukaryotic cells. And we engineered a plasmid that contained a TnpB gene and encoded rare RNA that targeted 5 different sites in the human genome. And again, using deep sequencing, we were able to demonstrate that TnpB was able to introduce indels at 3 out of the 5 sites with high efficiency. And in some cases, the efficiency was close to 20, quite high, close to 20% without any modification of TnpB protein or RNA sequence. So, in fact, these experiments demonstrated that TnpB protein emerges as a new genome editing tool. And it's, in fact, the smallest genome editing tool known to date. The size of TnpB is around 400 amino acid residues, and it requires a single rare RNA molecule as guide RNA. And importantly, it also functions as a monomer, because many of the Cas12 F family nucleases, they function as dimers. And TnpB requires, in fact, 5' TAM sequence for double strand break. So this brings me to my summary slide. And actually, during my talk today, I will show you that diving into natural diversity of Cas9 orthologues, we were able to identify Cas9 orthologues that show very different PAM requirements. So instead now of having a single pair of scissors, you have now multiple scissors that can cut DNA at different locations. In the next part of my talk, I showed you that actually using the same natural diversity approach, we were able to identify miniature nucleases. And finally, I showed you that actually we were able to identify also TnpB protein that is smallest RNA-guided nuclease known to date. And this TnpB protein is in fact not related to CRISPR, but is part of the transposon. And taking together all these tools actually make a really nice platform for developing very, very different tools for targeted genome modification. And finally, I would like to acknowledge people in my lab at Vilnius University, the team at Cas9 company, and our long-term collaborators at Corteva Agrisciences, New England Biolabs, Cardia, and thank you very much for your attention.
Well, thank you, Dr. Chiknis, for the insightful messages, especially on the opportunities and the specific challenges that needs to be overcome in CRISPR-Cas9 technology. Future development. CRISPR technology is especially an innovative technology compared to the other genome editing technologies, which won the recent Nobel Prize as well. Based on this technology, many pharmaceutical companies are developing therapeutics for incurable diseases, and this has brought many changes not only in the biopharma sector, but in genetic engineering field as a whole, including the agriculture sector, just like Dr. Chiknis said, in plant genome editing as well. With the lingering image that is left after Dr. Chiknis's keynote session, we would like to continue on with the session after a 20-minute break, after changing the stage. And as the next session will spot more on the future of genome editing technology with the renowned companies Cas9 and Cas12, along with the fireside chat with the industry leaders in genome editing, with Dr. Chiknis joining the panel as well. Thank you again for joining the keynote session with Dr. Chiknis at the third day of BIOPLUS-INTERPHEX Korea 2022. Please stay tuned.
Thank you.